Dear students, today in this lecture we are going to develop some understanding regarding the differences between prokaryotes and eukaryotes. At the leftmost part of this slide, we have documented features, and after the features we have prokaryotic cell and eukaryotic cell. So a comparison will be drawn in this lecture regarding prokaryotes and eukaryotes. The first feature is plasma membrane. Plasma membrane is invariably present all through the animal or living system of this planet, barring the viruses. So plasma membrane is present in both prokaryotes and eukaryotes. Come to the cell wall. It is non-cellulosic in prokaryotes and consists of amino sugars and muramic acid. Cell wall is totally absent in animals, and in eukaryotes, if it is present, it present only in plants, and it is cellulosic in chemical nature. The capsule is present in prokaryotes, and it is of mucopolysaccharides, but capsule is absent in the eukaryotic cell. Golgi apparatus. I have told you at the beginning of this series of this series of lecture that all the membrane bound organelles, they are wanting in prokaryotes. So the Golgi apparatus is also absent in prokaryotes, whereas it is present in eukaryotes and is associated with the cell secretion. The endoplasmic reticulum is also absent in prokaryotes. It is present in eukaryotes and is associated with synthesis and cellular transport. Ribosome, another, it's an non-membrane bound organelle, and it is 70 Swedberg type of ribosomes. They are present in prokaryotes, whereas 80 Swedberg types present in eukaryotes. In eukaryotes, the ribosomes, they may be associated with endoplasmic reticulum. Such an endoplasmic reticulum is designated as rough endoplasmic reticulum. Also, the ribosomes, they are laid free in the cytoplasm. However, the rough endoplasmic reticulum is not present in ribosomes. So the 70S type of ribosomes, which is the striking feature of these prokaryotes, is they are present in cytoplasm. Mitochondria, they are absent. The performance of functions of mitochondria is done by mesosomes, which is the inpushing of the plasma membrane. Mitochondria is present in eukaryotes, and it is associated with the cell respiration. The cell respiration is done in prokaryotes in the mesosomes. Microtubules, which are made up of tubulin proteins, confers the skeleton to the cell, are absent in the prokaryotes, present in cytoplasmic material in eukaryotes. The nuclear membrane is absent. Nuclear membrane is present around the nuclear material in eukaryotes. The nucleolus is also absent, and it is present in the eukaryotes. The lysosomes, they are absent, since lysosomes are also membrane bound organelles. They are present in eukaryotes and have digestive role to play. Vacuoles are again absent in prokaryotes, but are present in eukaryotes, but only in plant cells. They are absent in animals. The respiratory enzymes are located on plasma membrane, and it's in pushing, as I've told you in the early part of this lecture, at the mesosomes. The respiratory enzymes are enclosed in mitochondria. Photosynthetic apparatus is present in form of membranes with chlorophyll A in blue-green algae and bacteriochlorophyll in bacteria, whereas chloroplast contains the photosynthetic apparatus with chlorophyll A and B are present only in plant cells. Animals don't have chloroplast or photosynthetic machinery. Chromosomes. The prokaryotes have single circular DNA, which is not associated with histone proteins, but in place of histone proteins, they have polyamines. Eukaryotes have more than one chromosomes. They are associated with histone proteins and are enclosed in nuclear membrane. They have primitive method of division, the prokaryotes, and cell divides by fission, no mitosis, no meiosis. However, an elaborate system of mechanism of cell division is present in eukaryotes, and it is called as mitosis and meiosis. Flagella, if present, they have 9+0 microtubular arrangement as divorced from 9+2 microtubular arrangement in eukaryotes. The examples of bacterial cells are bacteria, blue-green algae, the pleuropneumonia-like organisms or mycoplasmas, the spirochetes and rickettsias. However, from protozoa to algae, all the metazoans and metaphyta, all of them, they are placed under eukaryota. Now let us have some sense of difference between a plant and animal cell. The cell wall is cellulosic in plant cell, present outside the plasma membrane in all the plants except algae. The cell wall is absent in animal cell. Plasma membrane is the outermost covering, so no cell wall in animals. Vacuoles present throughout the plant kingdom but absent in animals. The membrane of vacuole is called as tonoplast. Chloroplast present in plants but absent in animals. Centrioles and centrosomes are also absent in plants. In fact, they have microtubular organizing centers in place for the division, whereas centrioles, centrosomes are present in animal cells. Golgi called dictyosomes found scattered in the plant cells, whereas well developed it lies near the nucleus in animal cells. So students, this is all in this lecture, and in the next lecture we'll take up the history of cell biology. However, somehow one important thing has been left out. I forgot to discuss the properties of the protoplasm in the last lecture. Protoplasm, as I told you in that lecture in which I had mentioned, protoplasm is a polyphasic colloidal system which exists in two states, which is called as sol and gel phase. And these sol and gel phase, they are interchangeable, and this property is called as phase reversal, and it is performed by the process of solation and gelation. If the protoplasm undergoes solation, that means it is going to get converted into sol state, and if it is undergoing gelation, that means the sol state is converted into gel state. And this is very important as far as the movement of the cell is concerned, and also in certain activities of the cells. Properties of the cell wall, as I have told you and mentioned right now, is the phase reversal, the solation and gelation. Sol part may undergo gelation and gel part may undergo solation. This is called as phase reversal, as most of the metabolic reactions takes place around the nucleus. There is a reason that temperature there remains high, keeping the protoplasm there to exist in sol state, and at the ectoplasm the reverse condition exists. That means the ectoplasm largely remains in the gel state, whereas the protoplasm, which is in the vicinity of the nuclear membrane, tends to remain in gel state owing to the raised temperature which is caused by the reactions which are occurring around the nuclear membrane. It shows Tyndall effect, that means the scattering of the narrow beams of light by particles of dispersed phase. Other properties which are displayed by the protoplasm is contractility and viscosity, and the refractive index of the cytoplasm is 1.4, which indicates that the cytoplasm is more or less homogeneous. And the chemistry of the protoplasm is that it contains oxygen as an element to the level of 65%, carbon 18%, hydrogen 10%, nitrogen 3%, calcium 2.5%, phosphorus 1.5%, and rest some other trace elements. If you compare the composition of plants and animal cells, water is 75% in plant cells and around 67 to 70% in animal cells. Proteins, plant have 2%, whereas animal cells have 15% proteins. Plants have very low fat content, 1%, and 13% of the total material of animal cell is fat. Carbohydrates, plants have high carbohydrate content, 20%, whereas animal cell they have only 1%. So students, this is all in this lecture. In the next lecture we will be taking up the history of cell biology. Thank you very much.